Welcome back, everyone. Today, we're diving into some pretty exciting research. It's a paper from Nature Aging that's, well, kind of shaking things up in the world of aging research.
Yeah, it's definitely turning heads.
It has to do with those pesky senescent cells.
Ah, yes. The cellular troublemaker.
Exactly. Now, typically, researchers have been focusing on developing drugs called senolytics to get rid of those senescent cells. Just wipe them out.
Right, the direct approach. Target and destroy.
But this study, they're exploring something totally different. They're using, you might call it a matchmaker peptide.
A matchmaker, huh? That's a new one.
I know, right? It's a pretty clever analogy. So maybe you can explain this. For those who haven't been following every twist and turn in the world of senescent cell research, why is eliminating these cells such a big deal?
Well, senescent cells, they're basically cells that have stopped dividing. Like they've hit the brakes on replication.
They've reached retirement age, but they're refusing to leave the office.
Something like that. The problem is they should be dying off, but they're not. They hang around, and as they accumulate, they start causing all sorts of trouble.
Trouble.
Oh, you name it. Wrinkles, age-related diseases, the usual suspects.
So getting rid of them could potentially slow down aging.
That's the hope. And for a long time, the focus has been on those senolytic drugs, the ones that directly kill senescent cells.
Right, the seek and destroy missions.
Exactly. But there's a catch. Those senolytics can have some nasty side effects.
How so?
Well, they can be a little indiscriminate.
Indiscriminate.
Yeah, they can't always tell the difference between a harmful senescent cell and a perfectly healthy cell. So there's a risk of damaging the good guys along with the bad guys.
Kind of like trying to eliminate weeds on a flamethrower. You might get rid of the weeds, but you'll probably scorch your lawn in the process.
Yeah, that's a good analogy. So this new approach, this matchmaker peptide, it's trying to do things a bit differently.
So less flamethrower, more like a pair of tweezers carefully plucking out the bad cells.
Exactly. This approach is all about helping the body's own immune system do the heavy lifting. And see, the immune system is designed to recognize and eliminate damaged or abnormal cells. But sometimes, those senescent cells manage to fly under the radar. They're like those sneaking criminals who blend into a crowd.
So they need to be pointed out.
Right. And that's where this matchmaker peptide comes in. It's called E16-UPA24. It's like a homing beacon that seeks out a specific receptor found on senescent cells called UPAR.
Wait, so there's a specific marker on these senescent cells that this peptide can latch onto?
Yep, that's the key. And what's interesting is that this UPAR receptor is much more common on senescent cells than on healthy cells.
So it's like a unique identifier, a sort of fingerprint for these aging cells.
You got it. And the E16-UPA24 peptide, it's like a sniffer dog trained to sniff out that specific scent.
So it finds the senescent cells. Then what? Does it just signal the immune system like, hey, come over here, we've got a problem?
It's actually a bit more involved than that. It does more than just signal. It actually modifies the surface of the senescent cell.
Modifies? How so?
It attaches a molecule called polyglutamic acid to the cell.
Polyglutamic acid?
Yeah. Think of it like attaching a giant eat me sign to the senescent cell.
Okay, now that's a visual I won't soon forget. So this polyglutamic acid, it's basically a giant neon sign that attracts the attention of the immune system's cleanup crew.
You got it. It specifically attracts natural killer cells, or NK cells for short.
Natural killer cells? They sound pretty serious.
Oh, they are. They're like the body's elite soldiers. They're highly trained to identify and eliminate cells that are causing trouble.
So instead of directly killing the senescent cells, this peptide is basically flagging them for destruction by the immune system.
Exactly. It's like setting up a blind date between the senescent cell and the NK cell, and letting nature take its course.
That's pretty ingenious, but how did they even figure this out? Like, how do you go from senescent cells are a problem to let's attach this specific molecule to attract NK cells? It seems almost too clever to be true.
Well, it's a testament to the power of scientific curiosity and ingenuity. They didn't just stumble upon this solution by accident. It involved a lot of careful research and experimentation.
So walk us through it. What was some of the key steps that led to this discovery?
Well, they didn't just jump straight into testing on animals. They started with something called in silico experiments.
In silico?
Yeah, it basically means doing experiments in a computer simulation. They were able to model how the peptide would interact with both the senescent cells and the immune cells.
So they built a virtual world to test their theory before moving on to real-world experiments.
Right. They wanted to make sure their hypothesis held up in a controlled digital environment before they started testing it in living organisms.
That's a smart approach. So what did they find in these simulations? Did their virtual experiment work?
It did. The simulations showed that the peptide was very effective at binding to the UPAR receptor on senescent cells. And, even more importantly, it showed that this binding led to the recruitment of NK cells.
So their theory was working, at least in the digital world. But the real test would be to see if it worked in a living organism.
Exactly, and that's when they moved on to testing in mice.
Ah, the mouse model, a classic in scientific research. So, did this matchmaker peptide live up to the hype in the real world?
It did. They tested it in a bunch of different mouse models of aging and age-related diseases.
Right.
Well, they looked at liver fibrosis, lung injury, even cancer. And in each case, they found that the peptide, this E16UPA24, significantly reduced the number of senescent cells.
Wow, that's really impressive. It wasn't just effective in one specific condition. It seemed to work across the board.
Exactly. And what's even more exciting is that they didn't just observe a reduction in senescent cells, they actually saw improvements in the mice's overall health and function.
So, like, what kind of improvements are we talking about here?
Well, they used a variety of tests to assess the mice's physical abilities. One of them is called the road rod test.
Rotarod, what's that?
It's basically like a little rotating cylinder, kind of like a treadmill for mice. They put the mouse on the rod, and as it starts spinning, the mouse has to walk to stay on top.
Oh, I get it. So it's a test of their balance and coordination.
Right. And the mice that were treated with the peptide, they were able to stay on the road rod for a significantly longer time compared to the mice that didn't receive the treatment.
So they were more agile, more sure-footed?
Exactly. They also tested their grip strength, and again, the treated mice showed a marked improvement.
Wow, that's pretty compelling evidence that this peptide is doing something beneficial. It's not just about getting rid of the senescent cells, it's actually translating to a better quality of life for these mice.
Right. It's improving their physical function, which is a really important aspect of healthy aging.
Absolutely. I mean, what good is living longer if you can't enjoy those extra years?
Exactly. And that's what makes this research so promising. It suggests that we might be able to develop therapies that not only extend lifespan, but actually improve health span as well.
Health span, that's a great term. It's not just about adding years to your life, it's about adding life to your years. But before we get carried away with dreams of running marathons in our 90s, I have to ask, were there any downsides to this peptide? Any potential side effects?
Well, that's always a crucial question, right? And to their credit, the researchers did a very thorough safety assessment.
And what did they find?
They didn't observe any significant adverse effects in the mice.
So no red flags?
None so far. Now, of course, we can't extrapolate those results directly to humans.
Of course not. Mice aren't people.
Right. But the fact that they didn't see any obvious problems in the mice is definitely encouraging.
It's a good sign. But I'm curious, when they were looking at the effects of this peptide on the immune system, did they notice anything besides the increase in NK cells? Did it impact other parts of the immune system as well?
Yes, actually, they found that the peptide seemed to have a sort of calming effect on the immune system as a whole.
Calming.
Yeah. They noticed a decrease in certain types of inflammatory cells, which are often associated with tissue damage and the aging process in general.
So it wasn't just about boosting one type of immune cell, it was about creating a more balanced immune environment.
Precisely. It's almost like the peptide was acting as a conductor, making sure that all the different parts of the immune system were working together in harmony.
That's a great analogy. It really highlights the complexity of the immune system and how this peptide seems to be working in a very subtle and sophisticated way. It makes you wonder what other possibilities this type of approach might open up.
Absolutely. This research has really sparked a lot of excitement in the scientific community. It suggests that we might be able to harness the power of the immune system in a very targeted and precise way.
Imagine if we could develop similar therapies to target other harmful cells in the body, like cancer cells or cells involved in autoimmune diseases.
Exactly. The possibilities are really quite amazing.
It's mind-blowing to think about, but let's be realistic here. How far away are we from actually seeing something like this being used in humans? I'm sure there are a lot of hurdles to overcome before this type of therapy could become widely available.
Oh, absolutely. It's important to be cautious and not get ahead of ourselves. There's still a lot of research to be done before we can say for sure whether this approach will be safe and effective in people.
So, what are the next steps? What kind of research needs to be done?
Well, they'll need to conduct more expensive preclinical studies, test it in other animal models, figure out the optimal dosage, how to administer it, and then, of course, they'll need to carefully design and conduct human trials.
Right, those are all crucial steps. It's a long road from promising results in mice to an approved therapy for humans.
It is, but I'm optimistic. I think this research has laid a very strong foundation for future investigations, and I think it's only a matter of time before we start seeing more therapies that take advantage of this type of targeted immune modulation.
It's an exciting time to be alive, that's for sure. To be living in an era where we're making such incredible strides in understanding aging and how to potentially slow down or even reverse some of its effects. This study definitely gives us a lot to think about, and I'm sure our listeners are already buzzing with questions and ideas. But before we wrap things up, there was one aspect of the research that really caught my attention. You mentioned earlier that the peptide specifically targets the UPAR receptor, and that this receptor is much more common on senescent cells compared to healthy cells. Did they delve into why that is? What makes this receptor so prevalent on these troublesome cells?
That's a great question, and it gets to the heart of what makes senescent cells so unique. You see, UPAR isn't just a random receptor sitting on the cell surface. It actually plays a key role in the process of cellular senescence itself.
So the very process that creates these troublesome cells also makes them more susceptible to this peptide. That seems almost too good to be true.
It is a bit of a lucky break, scientifically speaking. You see, as cells go through their normal life cycle, they're constantly dividing and replicating. But each time a cell divides, its telomeres, which are like protective caps on the ends of chromosomes, get a little bit shorter.
It's kind of like a biological clock ticking away inside our cells.
Exactly. And eventually those telomeres get so short that the cell can't divide anymore. At that point, the cell enters a state called senescence. Now, in some cases, this is actually a good thing. It helps prevent damaged or potentially cancerous cells from continuing to divide uncontrollably.
So it's like a safety mechanism to prevent those rogue cells from causing too much trouble.
Precisely. But as we age, those senescent cells start to accumulate, and they don't always get cleared away as efficiently as they should. And here's where UPAR comes back into the picture. As cells enter senescence, they start to express higher levels of UPAR on their surface.
So it's almost like those senescent cells are waving a flag saying, hey, I'm over here and I'm not doing so well.
That's a great way to put it. And thanks to this research, we now have a way to target those flag cells and help the immune system eliminate them safely and effectively.
This is groundbreaking stuff, really. It seems like we're starting to unravel the mysteries of aging at a very fundamental level. But I know what our listeners are thinking right now. What does this all mean for us? What are the practical implications of this research?
Well, it's definitely exciting, but it's important to remember that this research is still in its early stages. It's going to take time, probably years, before we see any kind of therapy based on this matchmaker peptide available to humans. There are a lot of steps between, promising results in mice and a safe and effective treatment for people.
So don't expect to see this at your local pharmacy anytime soon.
Not quite. But I think this study opens up a whole new way of thinking about how we might approach aging and age-related diseases. It's a real paradigm shift.
Instead of just trying to manage the symptoms of aging, we're starting to think about actually targeting the very processes that drive aging at the cellular level.
Exactly. It's like we're moving from treating the disease to actually preventing it in the first place.
And that's got to be a game changer. I think that's what really resonates with people. It's not just about living longer, it's about living better.
Absolutely. And this research suggests that we might have more control over that than we thought.
It's kind of empowering, isn't it, to think that we might have a say in how our story unfolds, rather than just passively accepting the script that's been handed to us.
It is. And it's not just about individual empowerment either. Think about the implications for society as a whole. If we could delay or even prevent age-related diseases, it would have a huge impact on healthcare systems, economies, even our social structures.
It's almost overwhelming to think about all the potential ramifications, but I think it's important to acknowledge that there are also ethical considerations that need to be carefully addressed as we move forward with this type of research.
Oh, absolutely. Any new technology, especially one with the potential to so profoundly impact human health and longevity, it needs to be approached with a caution and thoughtful consideration of the ethical implications.
We need to make sure we're doing this right.
We do. We need to have open and honest conversations about the potential benefits and risks, and we need to make sure that these therapies are developed and used responsibly and equitably.
It's not just about the science, it's about the values that guide how we use that science.
Well said. We need to make sure that we're not just extending lifespan, but that we're extending health span and well-being for everyone.
Well, I think we've covered a lot of ground today, from the intricate workings of this matchmaker peptide to the broader implications of this research for aging and society as a whole. It's been a fascinating deep dive into the cutting edge of science, and I want to thank you for joining us on this journey and for sharing your expertise with us.
It's been my pleasure. Thanks for having me.
And as always, if you want to delve deeper into the science, be sure to check out the show notes for a link to the original Nature Aging article. Until next time, keep it curious.